Viable plate counting is a great technique for determining how many living cells are in a sample taken from the environment, somewhere out in the environment. Viable plate counts tell you the living cells that were in that original sample. We just have to remember that that means that if we compare that result to a result from a flow cytometer or a Coulter counter or even a microscopy count, the viable count is going to be less than the other counts, because the other counts will also count dead cells. Viable plate counts don't count dead cells. That's why viable plate counting is so useful sometimes. It's helpful for us to know how many living cells are present in a sample. The other thing to remember is this. Sometimes in an environmental sample, like a sample taken from, say, a recreational body of water, there's not going to be one kind of microbe growing in there. There's going to be multiple kinds of microbes. So the other thing that will affect our count is what kind of growth media that we use. A good example of this is, again, recreational bodies of water. We expect microbes to be growing in lakes and ponds and rivers. We expect that. There's nothing wrong with that. But we need to know, we need to monitor for any pathogenic microbes that might be present in that water. So what we can do is we can choose and auger a type of media that will only allow a certain subset of the microbes to grow, the pathogens. We're interested in the pathogens. We don't care about all the other microbes that aren't harmful to us. So viable plate counting can be used to grow a subset of all of the microbes that are in a sample. And that's a good thing, because it allows us to determine how many pathogenic microbes there are versus the total microbial count. Microscopy, cell counters, those would determine the total number of cells in there. So, viable plate counting is most useful to us when we use these very special kinds of growth media. We're going to be talking about these in detail in the next couple of weeks, but these types of growth media are called selective and differential media. They allow us to grow microbes very purposefully. They allow us to grow only the microbes we're interested in, the pathogens usually, and it prevents the growth of other kinds of microbes. So when we're sampling foods and water, even when we're doing cell counts in sewage at sewage treatment plants, we're often doing viable plate counting with selective or differential media because we're really interested in counting how many pathogenic organisms are present in that sample. Let's switch gears now, leave viable plate counting behind, and talk about, again, turbidity and how we can use turbidity in a sample to get an estimate of cell number in that sample. Remember, when you grow cells in a liquid broth, we're not talking about agar here, we're talking about liquid broths. As the cells start to divide, they're going to turn the broth cloudy. And we can actually measure how cloudy the broth has become after 24 hours of incubation. We're going to measure what's called the optical density of the broth. You need a machine called a spectrophotometer to do this. These are machines that are not particularly expensive. They're present in most laboratories, diagnostic laboratories, chemistry laboratories, biology laboratories. You find spectrophotometers in lots and lots of different laboratories. What a spectrophotometer does is it shines light through a sample, and depending on what's in the sample, like microbial cells, the light can be set at a particular wavelength so that it will be absorbed by the cells. That absorbance can be measured. We can then use the absorbance value to extrapolate back and calculate how many cells were in the original sample. This is an indirect method. It doesn't actually calculate cell number. It calculates optical density. We have to then take the optical density and convert it mathematically into a cell number. The higher the optical density, the more cells are in the tube. Now, doing a turbidity measurement, doing an optical density measurement, involves a little more math than what we saw with viable plate counting. You have to construct a graph first using a set of standards. That graph is called a standard curve, and it's from the standard curve that we're going to get an equation that's going to help us calculate cell number. The last technique I mentioned was polymerase chain reaction, or PCR. There are many different varieties of PCR available to us now in the laboratory, including what's called qPCR, or quantitative PCR. PCR is a technique where we place genetic material into a PCR machine, and that's what they're showing us here on the left side of the diagram. We place some DNA, for example, into the machine, and then the machine is going to amplify a particular section of that DNA. For example, a gene that we might be interested in. It's going to cycle through a process where the DNA in the area of that gene, shown in red, is going to get amplified. It's going to get copied again and again and again. And it's going to produce an exponential increase in the number of copies of that gene. So that by the end of many cycles, typically somewhere between 30 to 40 cycles through the machine, we're going to get millions and even billions of copies of that gene produced. Now, why would you want to amplify a piece of DNA like this? Well, maybe you want to study that gene. Maybe you want to know what the sequence of bases is in that gene. In order to do that, you're going to need a lot of copies of that gene for you to use in the laboratory. Sometimes we do PCR because we want to know if the gene is just present in a sample. For example, we've done a lot of PCR testing here on campus in order to monitor all of us for infection with SARS-CoV-2. We've all had the swab put up our nose and then sent off to the laboratory for PCR testing. What that PCR test is doing is it's looking for the presence of viral genetic material, a particular sequence of genetic material that only can be found in the SARS-CoV-2 virus. If it's present, the machine will amplify it, again, millions and billions of times, so it can be detected. In other words, PCR, by amplifying some piece of genetic material, will tell us if a particular microbe is present in the sample. But remember, we're not really interested in if, we're interested in knowing how much. How much? How many of this microbe? How many of these cells are present in a sample? In order to do that, you have to do a basic PCR that has been taken up a notch. You have to do what's called quantitative PCR. What quantitative PCR does is determine how many cells are present. So regular PCR tells you if that particular microbe is present. Quantitative PCR tells you how many cells are present in a sample. Very, very useful technique. Now, when we talked about growth in lecture in class last time, one of the things we talked about was that all bacteria, all microbes in general, are going to grow most effectively, most efficiently when they are in a certain environment. Every microbe has a particular temperature it likes to grow at. It has particular nutrients it likes to have available. It has a particular amount of light it likes to be in or out of. All microbes have specific growth conditions that they prefer. And out in the real world, when they're living in their normal environment, those conditions are usually met. And it allows the microbe to grow exponentially. Of course, once we come into the laboratory and we start to grow microbes, we have to try to mimic those perfect conditions as much as possible. If the conditions are not perfect, the cells won't grow exponentially for us. The types of conditions that impact growth include, well, many things. We talked about some of these in lecture. We talked about how temperature will control the growth of microbes. We talked about how putting a sample of food into a refrigerator will slow the growth of any microbial cells that might be present in that food. When we were in lecture, we talked about other conditions, things like pH, the level of acidity, or the level of alkalinity. We talked about several different conditions. What we're going to talk about today are a couple of other specific conditions. In addition to temperature, we're going to talk about water availability. Water availability refers not to the amount of water in a particular environment around a microbe, but whether or not that water is available for the microbe to use it. And finally, we're going to talk a little bit about salt, because salt, it turns out, is something we can manipulate in the laboratory in order to enhance the growth of certain microbes. I want to stress this fact that every single microbe has its own particular temperature that it loves to grow at. That's what we refer to as its optimum temperature. One of the things that we need to know when we're trying to grow microbes in the lab is what's known as the cardinal temperatures for a particular microbe. On this graph down here, you're looking at, on the x-axis, the temperature, and on the y-axis, the rate of growth. In other words, how fast microbial cells are dividing in the culture that we create in the laboratory. So the higher the temperature and the faster the growth rate. So take a look at the line on the graph. Over here at this low temperature, you can see that the growth rate is 0. That means that there's going to be a minimum temperature below which a certain microbial species just can't grow. It's just too cold for it to grow. Once we start going above that minimum temperature, if you follow the curve, the growth rate is going to start to increase as the temperature increases. It's going to go up and up and up and up and up until it hits a peak. There will be a temperature, one temperature, where you'll have the maximum growth rate for that particular microbe. That's the optimum temperature. If you get any higher than that, if the temperature gets any higher than that, the growth rate is going to drop off. And notice how fast it drops off. If you get above this temperature, you're not going to get any growth. This is the maximum temperature that that particular microbe can grow at. So every species of microbe, all the bacteria, the archaea, the microbe eukaryotes, They're going to have a minimum temperature for growth, a maximum temperature for growth, and an optimum temperature for growth. These are what are known as the cardinal temperatures. The minimum, the optimum, and the maximum. Those are the cardinal temperatures for that particular microbe. I want you to look at the labels that are on this graph as well. When you get down to the minimum temperature for growth of a particular species of microbe, at that low temperature, at that relatively low temperature, what's happening is the cell is just not functioning optimally, and that's why it can't grow. The membranes are getting cold, essentially. They're starting to get stiffer. They're actually starting to gel up. They're getting so stiff. Transport processes across the membrane slow down so much that the cell simply cannot grow at that moment. It cannot move things in and out of the cell fast enough to allow growth. As you start to warm things up, growth increases. And that's because things are getting better at the membrane. The membrane is starting to warm up. The transport processes that were happening very slowly before are starting to speed up now. The enzymatic reactions are occurring at increasingly rapid rates. When you get to the optimum temperature, things are just perfect for the cell. The cell is functioning optimally, so growth is occurring optimally. Now, when you get to the maximum temperature, it's warm enough now, or hot enough now, that the cell, again, cannot function normally. Proteins start to denature. They start to unfold and become non-functional. The cytoplasmic membrane becomes so warm and so fluid that it essentially collapses. Without a functioning membrane, the cell simply cannot survive at temperatures above maximum. So knowing these cardinal temperatures is really helpful for us when we're trying to grow specific species of microbes in the laboratory. Take a look at this slide. This slide again shows some cardinal temperatures. Again, temperature across the x-axis. Notice that it's in Celsius, not Fahrenheit. And then growth rate on the y-axis. Now you can see there are multiple curves on this graph representing multiple different species. Notice how varied the cardinal temperatures are. There's a particular type of archaea that is has a genus name of Polarimonus. This is an archaea species. Notice its cardinal temperatures. The minimum temperature is below 0. Remember, 0 is the freezing point on the Celsius scale. Its optimum temperature for growth is 4 degrees centigrade or Celsius. Compare that to this growth curve shown in green. This is one for E. coli, which is a bacterium. It has an optimal growth temperature of 39 degrees Celsius. That's about body temperature. It also has a pretty low minimum temperature and a relatively high maximum temperature. E. coli has a big range in its cardinal temperatures. Look at this next one. This one has a genus name of Geobacillus. This happens to be another archaea. Again, all of these temperatures are going up. The minimum temperature is higher, the optimal temperature is higher, the maximum temperature is higher. This particular organism just tolerates heat better than E. coli does or Polarimonus does. Here's another archaea that tolerates even higher temperatures. Here's another one that tolerates even higher temperatures, over 110 Celsius. Minimum temperature is 90 degrees Celsius. That's very hot. What you can see on this graph is the incredible range in cardinal temperatures that different organisms have. particularly the archaea. Remember, compared to the bacteria, the archaea are able to survive in a much greater range of environmental conditions than the bacteria can. The archaea have evolved to take advantage of extreme environments in some cases, including extremes of temperature. There are archaea that can survive in very cold temperatures. There aren't many bacteria that can do that. There are archaea that can survive in extremely hot temperatures. There aren't many bacteria that can do that. I want you to notice the terminology that's been applied to each one of these graphs, because this is terminology we use when we talk about archaeal species especially. Any archaea that is capable of growing in extreme cold temperatures is referred to as a psychrophile. If you translate this term, it means a lover of the cold. Now, most bacteria are what are called mesophiles. If you translate this word, it means they're lovers of middle range temperatures. A thermophile is an organism that loves heat. That's what thermophile means. A hyperthermophile is an organism that loves extreme heat. So most bacteria are mesophiles. Archaea can be mesophiles. There are some archaea that are mesophiles, but there are also some archaea that are psychrophiles, and some that are thermophiles, and some that are hyperthermophiles. Archaea, certain species of archaea, can tolerate extremes of temperature much better than bacteria can. We're going to switch gears here and talk about this water availability concept, because this is especially important when it comes to keeping our foods safe. Remember, it's not just the amount of water that is in an environment where a microbe is living. It's also whether or not that water is available to the cell. This relates to what's called osmotic stress. Remember, living cells are full of water. Living things are between 65 to 75% water. All microbes, like all cells, require water in order to survive. They are largely made out of water. But here's the thing that's important to remember. Water, because it's a polar molecule, is attracted to certain dissolved molecules. And when it becomes attracted to those molecules, the partial charges in all those polar things are going to be attracted and pull the molecules closer together. Take a look at this drawing over here. You're looking at a representation of a sodium ion in the center, and it's surrounded by 6 water molecules. Remember, water is polar. That means it's going to contain partial charges. The oxygen in the middle of the water is going to be partially negative. The hydrogen atoms are going to be partially positive. The partial charges form because in the covalent bonds, the electrons are being shared unequally. Oxygen has a higher electronegativity than hydrogen does. So it pulls the electrons closer to it, and it becomes partially negative. The hydrogens are relatively farther away from the electron pair, and so they are partially positive. This particular sodium ion, it's not partially positive, it's actually positive. It has a positive charge. It's an ion. It's what we call a cation because it has a positive charge. In an environment where there are sodium ions, Water molecules are going to be drawn to that sodium because the partially negative oxygens will be attracted to the positive ion. And what happens is this sodium becomes surrounded by water molecules. We refer to it as being hydrated. The sodium ion has become hydrated. This sodium, because it's surrounded by these these water molecules, this sodium isn't really available right now because all these waters are surrounding it, attracted to it. It's not really available to a microbe right now. Neither is the water. The water is essentially too busy being attracted to the sodium to be available to the microbial cell for use. This water is not going to pass into the cell and do jobs for the cell because it's busy. It's busy being attracted to the sodium. There are lots of different molecules in the environment that can become hydrated like this, that can essentially keep the water busy so it's not available to the microbe. Now, the other thing I've written on the slide is just a reminder to us that the kinds of substances that are outside of and in the environment of a microbial cell can also impact whether osmosis occurs. Remember, osmosis occurs when there's a gradient of a particular type of molecule, so a concentration difference between the inside and the outside of a cell, and that molecule, for whatever reason, cannot travel across the membrane to get rid of the gradient. Osmosis is going to occur. It's going to occur to try to equalize the concentration. If the molecule can't travel across the membrane to equalize its own concentration across the membrane, water will respond. So we really have two issues here. We have this issue of water being associated with dissolved substances. We have this issue where, depending on the concentration of substances in and out of the cell, we can get osmosis occurring. This is all affecting how the cell is existing, functioning, and again, whether or not all of this water is even available to it.